So today I'm going to present about the methods in gene cloning. Basically, there are 5 methods. The first method is isolation of gene. The second method is cleave or cutting the DNA. The third method was insertion. The fourth method was transformation and amplification. And the last method, which is screening. What is the isolation of gene? Basically, the isolation of gene is the process of which we need to choose the bacteria and choose the target gene from the wanted The first method was isolation of gene. What is isolation of gene? Isolation of gene is the process where we choose a bacteria. There are many types of bacteria, and the mostly common use was E. coli, and we choose the DNA fragment, which is a DNA fragment that choose was the target DNA. Basically, like if we want to make an insulin, we need to choose a target DNA which have insulin. Next, after we choosing all of this, we need to cut the DNA fragment from the DNA that we choose, and the bacteria cell from the bacteria that we choose. The next process is cleave or cut. Basically, there are 2 types of things that we need to cut. The first thing, we need to cut the things from the DNA, and we also need to cut from the plasmid. Basically, the plasmid, which is the bacteria cell, we only need to cut one time, which is when the bacteria can be processed many bacteria after we cut. But in the DNA, we need to cut many times because it can't be processed like the bacteria does. So, after we cut, the restriction enzyme that used for the cleave and the cut process was the same. We need to use the same restriction enzyme. If in the test tube A for plasmid, we use EcoR1, then in a test tube B for DNA, we need to use EcoR1 because the same restriction enzyme can produce the same types of end, which is the sticky ends for EcoR1, and which is that it was good and can be used because of the complementary. Next, I would like to continue to the third step, which is the insertion step. The insertion step is the step which of the DNA fragment will be inserted to the plasmid. Why it was being inserted? It will be inserted to produce the recombinant plasmid and non-recombinant plasmid. The target was we want the recombinant plasmid, but we also produce non-recombinant plasmid, so we can separate it by the next process. But in this process, we need to cut some the plasmid by using the complementary base. The complementary base is important because we can produce the DNA ligase. DNA ligase will act as a enzyme that will seal the plasmid so that it will form the phosphodiester bond. The phosphodiester bond is very important in this process. On the next process, we will look at the process of transformation and amplification. What is transformation? The transform words is the process of the bacteria which is we use as a E. coli. E. coli will be removed and will be added insulin from the DNA. The DNA fragment that were added inside the E. coli is the process called transformation. Why we need to add the insulin from the DNA that we took from the DNA? This is because the bacteria cells can't make its own insulin. So that is why the words of transformation will be produced, and the transformation will produce 3 types of bacteria cell, which is we produce 3 types of plasmid. The first plasmid is the plasmid that with recombinant, the plasmid with recombinant DNA, the plasmid that we wanted the most. The second is the plasmid without the non-recombinant. It means that the plasmid is self-ligated. It was non-recombinant. The non-recombinant plasmid, also known as a self-ligated plasmid. The third plasmid that produced was the plasmid that doesn't deserve or doesn't accept any other plasmid, which is we only wanted the recombinant plasmid. The recombinant plasmid was very important, and then after that the 3 types of plasmid were produced, the plasmid will be amplified with the amplification, and it will form a lot of colonies, which is the colonies also form through the recombinant plasmid, the non-recombinant plasmid or self-ligated plasmid, and also the plasmid that have non-plasmid. There was 3 types of DNA that produced. First, the DNA with a recombinant plasmid, which is the DNA that we wanted the most. The second is the DNA without the non-recombinant plasmid, which is the self-ligated DNA. And the third one is the DNA without any other plasmid. Lastly is the process of screening. There are 3 types of screening, which is antibiotic, the blue-white screening, and the nucleic acid prohybridization. The antibiotic was the screening that will produce 2 types. The DNA will be dissolved in the petri dish that contain of antibiotic. If the DNA have ampicillin resistance, it will grow, means it will be more. It will become more and a lot of DNA, a lot type of DNA. That means it is a recombinant DNA that we wanted. If the DNA will not grow, it means that it have no ampicillin resistance, which is it have no AMPR. The DNA have no AMPR, and the answer was the non-recombinant DNA. So the non-recombinant DNA is not that we want. So if in the petri dish we only have this, that means this is DNA that we wanted. So this is the recombinant DNA. That's how we choose using the antibiotic process. Next is through the blue-white screening. What is the blue-white screening? The blue-white screening is the DNA that produced will put in a petri dish. When we will put in a petri dish, there are contain of the lactose, which is galactosidase in the petri dish. If the DNA become blue, that is means that inside the DNA was the non-recombinant. Why is it non-recombinant? Because the DNA contain of LACZ gene and it will encode it with the galactosidase. That is why it become blue. So that is not the DNA that we wanted. So if we saw the blue DNA, we will remove it directly. But if the DNA was recombinant DNA, it will become white. That means that is what we wanted. Because what is become white? Because it cannot hydrolyze in the LACZ gene and in the galactosidase in the petri dish, so it will cannot rise with X-gal, which is the lactose, these things, the lactosidase. And this is the recombinant DNA, and this is the DNA that we wanted the most. Lastly is a nucleic acid prohybridization. What is nucleic acid prohybridization? This is a very, very simple process because this process was the high in technology. So in this nutrition plate, it will located all of the DNA, and the DNA will be in the nylon membrane. The nylon membrane will be wrapped with a plastic, and when it was wrapped in a plastic and it will cover with a nylon shim membrane, it will become the unwanted DNA, which is the non-recombinant, and the DNA without plasmid. It will be destroyed. So the only DNA that left by using this technology is recombinant DNA. So this is a very simple yet the very complicated and the very easy types of screening that we can use. So that is all for today. The conclusion is we have five methods in overview, which is the isolation where we choose the types of DNA and the type of bacterial cell, and the cleave or cut using the restriction enzyme. We need to make sure that the restriction enzyme we used was the same. And the next step is the insertion, which the DNA fragment, which is the insulin, the DNA fragment that we wanted, will be inserted into the plasmid of the bacterial cell. And the fourth step is transformation. We will just transform of the E. coli to become E. coli with insulin DNA, and after that the insulin DNA will be amplified with a lot of colonies of the types of DNA that produce, which is have 3 types. DNA with the self-ligated DNA with the recombinant, and the DNA that have no ligated at all, no plasmid at all. And last type is the screening. Screening is the process that we choose types of DNA that we want, which is the recombinant DNA, and this is the type, and this is how we can determine the type of DNA, and we can produce a lot of recombinant DNA that we wanted. So that's all for me. Thank you.